Welcome to part 2 of online lecture 5. In this lecture, we're going to discuss DNA replication, which you can read about in section 9.2 of your textbook. As we saw in chapter 7, the process of DNA replication occurs during the S phase of the cell cycle, which in eukaryotes precedes the division of the nucleus, or mitosis. DNA replication occurs in what's called a semi-conservative fashion. And what this means is that during DNA replication, the two strands are actually separated from each other or denatured, and each one of these strands serves as the template for a new strand. So after one round of replication, the two new strands that result consist of one of the original parent strands and one of the new daughter strands. This arrangement is shown on the bottom of your screen. In number 1, you can see the original double-stranded alpha helix, and both of the strands are shown in red. That red coloration out the bottom picture down here indicates an original parent strand. In step 2, the strands are separated and new bases are added using complementary base pairing, so that in step 3, the final step, the two new daughter strands that have resulted from this process each consist of one parental strand and one of the daughter strands. Now, the evidence for this semi-conservative replication of DNA actually came from a series of experiments using radioactive labels. The process of DNA replication begins when a complex of proteins binds to the DNA. This complex of proteins is called the pre-replication complex, and it doesn't bind just anywhere on the DNA. It binds at a very specific location called the origin of replication. In the bottom of your slide here, which shows a prokaryotic chromosome, the origin of replication is shown in pink. And whenever you see this ORI abbreviation written and italicized, that lets you know that that indicates the origin of replication along a chromosome. This complex that binds to the origin of replication, as I said before, it's very large, so it contains quite a number of proteins. But one of the most important ones is a protein called DNA polymerase. Hopefully by now you recognize that the ASE suffix actually indicates an enzyme. And usually you can tell by looking at the name of the enzyme what the function is. So a DNA polymerase is an enzyme which builds DNA polymers. When the pre-replication complex binds to the origin of replication, it actually causes the DNA to unwind. There's one specific enzyme called helicase, which is part of the pre-replication complex. And helicase is responsible for this unwinding. When the pre-replication complex binds to the origin of replication and the DNA unwinds, replication proceeds in two different directions. We use the term replication fork to describe the site where the DNA is unwound. The example that we just looked at was one of a prokaryotic chromosome. And you saw from that slide that prokaryotic chromosomes have only a single origin of replication. Well, eukaryotes have very different chromosomes. They're linear, and they have very many chromosomes. In fact, in each one of your cells, there are 23 pairs of chromosomes, or 46 individual chromosomes. Again, these are linear, and they're very long. And it would take a very long time for these chromosomes to replicate. So unlike what we saw in prokaryotic chromosomes, eukaryotic chromosomes have multiple origins of replication. On the right-hand side of your screen here, you can see a full set of human chromosomes. There are 22 chromosomes that are not involved in sex determination. Those are called the autosomes. And then there are 2 sex chromosomes that are shown over here, the X and Y chromosomes, and those are involved in determining sex. Everywhere you see one of the red boxes on a chromosome indicates an origin of DNA replication. And you can see that there are many more than there were in the prokaryotic cell. And the advantage of having all of these origins of replication is that it dramatically speeds up replication. And this is sort of showing you how this set of chromosomes would actually be replicated, again with each box indicating an origin of replication proceeding in both directions. This extra number of origins of replication in eukaryotes is vitally important. Compared to prokaryotic genomes, and a genome just refers to all of the DNA in a cell, but compared to prokaryotic genomes, eukaryotic genomes have about That's 100 times as much DNA as E. coli. And generally speaking, the rule of thumb for DNA replication is that the replication process proceeds in a manner so that there's about 2,000 base pairs being replicated per minute. That's pretty fast, actually, for an enzyme. But think about this. Human beings have about 3 billion base pairs of DNA in each cell. And so if there was only a single origin of replication, it would take about 2.6 years to replicate the genome. That won't work to serve the needs of a growing and reproducing organism. Can you imagine if it took 2.6 years to regenerate every skin cell in your body? So obviously these multiple origins of replication are very important, and in that they allow the genome to be replicated in just a few hours. So what happened is that when DNA replication occurs at these many different forks, the forks basically collide when they come together, and you end up with a long continuous chromosome. The slide at the top of your screen indicates with the red arrows multiple origins of replication in eukaryotic DNA. So it's very interesting. So even though eukaryotes have much larger genomes, they actually replicate faster than prokaryotes, which have smaller genomes. So for example, Drosophila takes about 3 minutes for its entire genome to replicate, even though it has about 100 times as much DNA as E. coli, whereas E. coli takes about 30 minutes. Now don't confuse this with development. Eukaryotic cells still develop much more slowly than prokaryotic cells because in addition to this replication of the DNA, there are organelles and lots of other complex cytoskeletal structures that actually have to be duplicated. But the DNA replication process itself is actually sped up dramatically by these multiple origins of replication. Now we're ready to focus on the process of DNA replication. This slide gives you an overview of the mechanism of DNA replication with particular focus on all of the enzymes that are involved in the process. We're going to go over each one of these steps in great detail, but I think it's helpful at the outset to get an idea of all the steps that are involved in DNA replication so that as we go through and go over each individual step, you already have the whole picture in your mind. There are 7 enzymes involved in DNA replication, and here are their jobs. The first enzyme is DNA polymerase 3. It's shown in this slide as the green blob here. Its job is to add new nucleotides to the growing strand of DNA using complementary base pairing as a guide. The second enzyme is helicase, which is shown here in this cream color. The job of helicase is to unwind the DNA ahead of the DNA polymerase so that the bases are exposed and available for replication. There's another group of proteins called the single-stranded binding proteins, and they're indicated by these little sets of 4 purple dots arranged in a square. They bind to the denatured or single-stranded DNA and keep it from reforming another double helix with the other parent strand instead of being replicated. The next set of enzymes are the topoisomerases. They're not shown in this picture, but they have a very important job, and that is that they relieve the strain that is induced in the double helix when helicase unwinds it. You can sort of think about this if you were to take a twisted piece of yarn and actually start pulling it from the bottom and separating the 2 strands of yarn. You know that the pieces of yarn that were still wrapped around each other would actually get tighter. So that's the strain I'm referring to, and the topoisomerases actually relieve that. The next enzyme is an enzyme called primase. It is responsible for synthesizing an RNA primer, which actually provides the nucleotide with the first 3' hydroxyl group that a DNA polymerase attaches a nucleotide to. This might be new news to you that RNA is used in DNA replication because we know if we look at a chromosome, we don't see that it's full of lots of little bits of RNA. So those bits of RNA are removed. That is the job of an enzyme called DNA polymerase 1. And DNA polymerase 1 removes the primer and replaces those RNA bases with DNA bases, but it's not able to completely fill the gap. It leaves very small gaps of about 1 base pair, and is the job of the last enzyme on this list, an enzyme called DNA ligase, to patch up these small single nucleotide base pair gaps that are left when DNA polymerase 1 is finished removing the primer and adding bases. With our understanding of the enzymes involved in the process, we're ready to turn our attention to an overview of the mechanism of DNA synthesis and of all the steps that are involved in that process. We're going to cover each of these in detail, so this is just meant to give you an overall introduction at this point. The first step occurs when helicase binds to the DNA, unzips a small region of it, forming what we call the replication fork. And this happens with topoisomerase binding out ahead of the replication fork to relieve that supercoiling we just talked about in the previous slide. Right away, the single-stranded binding proteins bind to keep the DNA from renaturing. RNA primase then makes a primer, and then DNA polymerase uses the 3' end of this primer to add nucleotides to it, using complementary base pairing as a guide. On one strand, this replication actually proceeds in the same direction as the replication fork is moving. That is the strand that we refer to as the leading strand. On the other strand of DNA, the DNA replication actually takes place in the opposite direction of that in which the replication fork is moving. We call this strand the lagging strand because DNA replication is slower on this strand. And you're going to see that what happens on this lagging strand is that DNA synthesis has to start over again and again with the synthesis of new primers, which slows down the process. And of course, this direction of replication occurring in one case towards the fork and the other case away from the fork arises specifically from the anti-parallel nature of the two DNA strands. Finally, DNA polymerase 1 comes along and removes the RNA primers, fills in most of the nucleotide bases that are left, except for one, and DNA ligase comes along and patches that little gap. Let's take a detailed look now at the process of DNA replication. As I said previously, the first step is the unwinding of the two parent strands of DNA and separation of the hydrogen bonds between them. This is catalyzed by an enzyme called helicase. The actual replication process begins with a short primer. This is a short bit of RNA that actually starts the process. RNA is synthesized by an enzyme called primase, and in the slide on the right-hand side of your screen here, you can see primase is indicated in this baby blue color. Our template strand is shown in the darker blue color, and then the primase comes along and actually synthesizes a short piece of RNA, which is shown in the teal color, that is complementary to this parent strand. Once the RNA primer has been synthesized, the DNA polymerase comes along and catalyzes the formation of phosphodiester bonds between the nucleotides. It does this one base at a time using complementary base pairing as a guide. And remember that this DNA synthesis, the reaction that's catalyzed by DNA polymerase 3, has directionality to it. Remember our nucleotides with their 5' phosphate and the 3' hydroxyl group? The synthesis is occurring in this way. Here's your existing nucleotide, and the phosphate group is being added to it. And that's why we call it a 5' to 3' synthesis, because the strand is growing in the direction where it started at the 5' end, and it's lengthening at the 3'. Let's take a closer look at DNA polymerase, the enzymes that are involved in catalyzing the synthesis of these phosphodiester bonds. These are really interesting enzymes. They're very big, and they have what we call a very high processivity. This means it's able to very quickly catalyze a large number of reactions in a fairly short period of time. The enzyme itself is shaped like an open right hand. So if you take your right hand out in front of you and point it in such a way that your thumb is pointing to the left-hand side of your body, you're sort of mimicking the way the DNA polymerase attaches onto the DNA. The palm part of your hand is where the active site is actually bringing the substrates into contact. And where your fingers are is actually the part of the DNA that recognizes the nucleotide bases and assists with the complementary base pairing. The enzyme catalyzes the reaction that's shown here on the bottom of your screen. We've talked about this over and over again, but it's really important, so I want you to get it. And that is that each nucleotide that comes in is actually added to an existing hydroxyl group. And once this phosphodiester bond has formed, that's a covalent bond, it's very strong, you'll see that there is the release of two of the phosphate groups that were initially attached to the nucleotide. Here's something that you should appreciate now that you've had our first exam and covered some of the topics in Chapter 2, and that's that the energy that drives DNA synthesis actually comes from the cleavage of this 2-phosphate molecule, which is called a pyrophosphate. This is a very high energy bond between the two of them, and it is actually the exergonic reaction that is coupled to the endergonic formation of DNA. I want to revisit this idea of the leading and lagging strand here. So the enzyme, at this stage of the process, the enzyme has actually bound to the DNA and has started to add nucleotides to the existing RNA primer that was synthesized by primase. And I mentioned in the overview that on one strand, the direction of DNA synthesis was going to move towards the replication fork. And you can see this here on the top of your screen on what we call the leading strand. And again, the difference in directionality on these two strands is just caused by the anti-parallel orientation of the two strands of DNA. When you unzip them, one is oriented in such a way that the 5' to 3' direction points towards the fork. On the other fork, the 5' to 3' orientation on a new strand points the opposite way that the fork is moving. So you can see from this slide that what's going to happen is that on the leading strand, one primer can be synthesized and DNA polymerase will continue to add nucleotides to it until it reaches the end of the chromosome. But on the other strand, on the lagging strand, once a primer is synthesized and the replication fork continues moving, you can see that DNA synthesis would stop unless there was synthesis of a new primer upstream of the original primer, and then DNA synthesis would have to proceed from that new primer. And this is in fact exactly what happens. So what happens on the lagging strand then is that DNA synthesis occurs in a series of small discontinuous stretches. And if you were to stop it at this point of the DNA replication process, what you'd see is that one strand, the leading strand, would have one RNA primer and a bunch of nucleotides added after that. And on the lagging strand, you would see a bunch of what are called Okazaki fragments. And these are short stretches of RNA primer that are followed by a bunch of nucleotides and then a gap between those nucleotides and the next RNA primer. These are actually named after a Japanese scientist. His name was Okazaki, and he actually made several brilliant discoveries, but he was unfortunately around Hiroshima when the atomic bomb was dropped, and he eventually died of leukemia at a fairly young age, in his 40s. So it would have been interesting to see what kind of contribution he would have made had he lived a little bit longer. Now, obviously, having this RNA in the DNA is a real problem, and we know for a fact that there aren't little bits of RNA in our chromosomes. And so there must be a mechanism for removing it, and that involves the two enzymes that we have discussed previously. This animation is showing you how this process actually works. On the top strand, you can see the leading strand proceeding in the same direction as the replication fork. On the bottom strand, you can see the Okazaki fragments being synthesized as replication is proceeding in the opposite way that the replication fork is moving. You can also see in this animation the RNA primers, which are indicated in green, and the DNA bases, which are indicated in red. And you can see that after the Okazaki fragments are synthesized, that DNA polymerase 1 is actually removing the RNA primer, and it's also filling in the bases. And finally, the gap is filled in by DNA ligase. So let's take a closer look at this process. So this is actually showing DNA polymerase where it has bound to the RNA primer. We know this primer is RNA because you can see that it has uracil instead of thymine. So DNA polymerase comes along and removes that RNA primer. It then fills it in with DNA bases, fills each gap with a nucleotide, and it continues to do this until it reaches the very last one. And when the primer removal is completed, DNA ligase actually replaces the DNA polymerase 1 at this very precise location where there is a single stranded gap. And DNA ligase then catalyzes the formation of a phosphodiester bond and then joins the Okazaki fragments. And at this point, the DNA synthesis process is complete. So let's summarize what we've talked about so far. These are all of the steps that we've talked about and all of the things I would like for you to understand about DNA synthesis. So first step, supercoiled DNA is relaxed by topoisomerases as helicase breaks the hydrogen bonds between the two strands. The single-stranded binding proteins then coat the single-stranded DNA to prevent it from renaturing. Primase synthesizes many primers on the lagging strand and only one on the leading strand. DNA polymerase III then synthesizes DNA in the 5' to 3' orientation by always adding a phosphate group to an existing hydroxyl group. It does it in a continuous fashion on the leading strand, and what I mean by that is that it doesn't have to start over and over again, and it does it in a discontinuous fashion on the lagging strand where it has to start over and over again. And of course those differences are caused by the different orientation of the two DNA strands. Finally, DNA polymerase binds to the RNA primers and removes them and fills in the gaps, all except for one, with DNA nucleotides. And then the one base pair gap that's left after DNA polymerase I does its job is actually filled in by DNA ligase. So this DNA synthesis is going to continue all the way to the end of the chromosome. And on the leading strand, this synthesis is going to continue until the end is reached and one RNA primer will be removed. But on the lagging strand, the presence of these RNA primers create a real problem. And that is that when the end of the chromosome is reached and the primer is removed, there is no upstream hydroxyl group for the DNA polymerase I to come along and add nucleotides to. So the bases don't get filled in. So what happens then is that in the absence of any other action, the chromosomes actually get shorter and shorter with each round of replication, somewhere between 10 to 40 base pairs. And you can see this poor little guy on the bottom of the screen. He's not very happy to hear that his chromosomes are getting shorter. Let's take a closer look at these ends of the chromosomes. They're actually referred to as what are called telomeres, and they have a very unique sequence. They actually have a series of bases that repeat over and over again. We call this a repetitive sequence. And these occur at the end of all eukaryotic chromosomes. And one of the things that's interesting to me anyways about these telomeres is that the sequence of bases that repeats over and over again is species specific. So yeast, for example, have different telomeres than do humans. So what's very interesting about these telomeres is because of that, what we call the end replication problem that we discussed in the previous slide, the loss of a little bit of DNA due to the removal of the primer through each round of replication, that a little bit of the telomere gets lost each time. But what's really interesting is that this only happens in certain cells. It does not happen in cells that divide continuously, like bone marrow cells or gametes. And by gametes, I mean sperm cells or egg cells. So what's happening in these cells that can actually continuously divide and don't have this penalty of the shorter chromosomes? Well, it turns out that there is an enzyme called telomerase that is active in these cells. And telomerase is very interesting. It actually contains a piece of RNA that actually acts as a template to add DNA sequences to the ends of these chromosomes to keep them from getting shorter. And we'll look at the precise mechanism for that in just a moment. But before we look at telomerase activity, I'd like to consider what's happening in all of the other cells, which are actually most of the cells in your body. So most cells in our body are not continuously dividing cells. And what happens in the other cells of your body is that they reach something that's called the Hayflick limit, or a fancier term for this is replicative senescence. And this was first described in a series of experiments by Pearl. And Pearl grew fibroblasts in culture, and they're just the cells that make up connective tissue. And what Pearl observed was that normally after about 50 cellular divisions, the cells just petered out. They stopped growing. They didn't die, but they didn't divide anymore. And this has actually been confirmed experimentally many times. And one thing that's really interesting is that the rate of cellular division actually slows down according to human age as well. So a young person's cells divide in culture more often than do those of older people's. So for example, a fetal cell can divide about 70 times, and in an older adult, then the cells only divide 10 or 20 times. And on average, we use around 40 to 50 cellular divisions for a typical cell. So cells cannot continuously divide. And there's one good reason for this. Although if you're someone who is experiencing the aging process, you might not agree with having your cells sort of peter out after a while being a good thing. But if we think about how cancer works, and cancer is actually a disease of the cell cycle gone wrong and cells dividing out of control, that if a cell only has a certain number of divisions associated with it, then a cancer cell would actually eventually run out of DNA, right? Well, not exactly. It seems like that telomeres may actually serve as sort of the genetic fountain of youth in cancer cells. And cancer cells, by definition, are what are called immortalized. And you may be familiar with HeLa cells. Those were cells that were isolated from the tumor of an African-American woman at Johns Hopkins. And those cells were really an amazing discovery as far as science goes, because it was the first time folks were able to grow human cells in tissue culture and do experiments with them. And the reason that they were able to keep growing those cells, we call that growing a cell line, indefinitely, is that those cells had become immortalized. And so the question is, how does that actually happen? So how is it that cancer cells are able to escape this Hayflick limit and actually become immortal? Well, cancer, first of all, actually refers to a multi-step disease that involves many different genes. So I don't want to oversimplify it here. But in many cancers, what has been observed is that telomerase has actually become active in cells in which it would typically not be. And so what's the big deal about this telomerase? Well, telomerase is what we call a ribonucleoprotein. That means it consists partially of RNA, or ribonucleotide, and also of some proteins. And it's a fascinating molecule with different functions. And this, the discovery of it was actually the basis for the awarding of the 2009 Nobel Prize. It's very interesting because the RNA part of telomerase actually serves as a template for the addition of DNA to the end of the chromosome, and the protein part of this enzyme actually catalyzes the formation of phosphodiester bonds. I don't want to go through the mechanism in an extreme amount of detail here. What's shown on the right-hand side of your screen is just what happens when telomerase binds, and then using its RNA bit as a template actually allows for the extension of one strand of DNA. In this case, you can see the strand growing in the 5' to 3' orientation. And what will happen when it grows far enough out, primase will actually synthesize a new primer on that strand, which will then be used to fill in some of the gap. So telomerase is active in both germline cells, so it's typically active in cells that give rise to gametes, and also in some stem cells in eukaryotes. But when it becomes active in cells where it shouldn't be, it can cause a lot of problems for the organism. For example, one of the most common mutations in cancers of all types occurs in a gene for an enzyme called TERT, or telomerase reverse transcriptase. And here you're looking at two different mice. The one on the left is 48 weeks old. The one on the right is only 35 weeks old, but TERT has become active in this mouse, and you can start to see the formation of some tumors on the back of this mouse. So this reactivation of TERT then can cause cells to become immortal, and of course a tumor is actually just a mass of cells that are growing out of control. So there are a variety of different diseases that are associated with telomerases. For example, there's a disease called Werner syndrome, and this is sort of the opposite of cancer. And in this case, telomerase is actually not active when it should be. And so what happens is that instead of having a normal aging process, in this Werner disease, there's actually an early onset of some of the features of aging. And you can look at these pictures of the woman on the bottom of the screen, and you can see by the time she's 48, she has Werner syndrome. You can see by the time she's 48, she looks like a much older person, maybe someone that would be in their 60s. And by the time she was 56, she actually looked like someone who'd be in their 70s or 80s. And we know the genetic basis of this syndrome, and it's actually it actually occurs not in the telomerase gene, but it occurs in a gene for a helicase that is required for telomerase activity. So the message, of course, then is that telomerase activity has a very powerful effect on the aging process of cells and on cancer, but also on the aging process of the entire organism. So it's important then that telomerase activity be controlled. It should occur in cells that need to continuously divide, but not in other cells. But without telomerase being active, it can actually artificially speed up the aging process. This concludes part 2 of lecture 5. Part 3 will cover DNA repair systems.